Welcome back to our discussion of the kidneys. I want to remind you that we have been talking about the four main processes that happen in the kidneys. So we've gone through them one by one, and now we are on our last one, excretion. And I'm going to also talk about a related concept called clearance. Okay, so this diagram shows the process of excretion. I want to remind you of our other processes that are happening in the tubule, right? So first we have filtration of fluids from the glomerulus into Bowman's capsule, okay? Anything that we want to keep in our bodies, we are going to reabsorb from the filtrate that is in the tubule. Any substances that we want to especially get rid of, we want to get rid of more of, we're going to secrete into the tubule. And then whatever ends up in the tubule at the end gets excreted from our bodies as urine, okay? Therefore, we have this mathematical relationship shown here. Excretion equals filtration minus reabsorption plus secretion, okay, if we're looking at a particular solute. All right, so urine, which is the substance that we excrete from our bodies, can be a very useful diagnostic because it is a reflection of all of these processes, right? It's the end product of all of these processes that have happened in the kidney. Therefore, it can tell us a lot about what happened in the kidneys and how it handled certain substances, okay? One way in which urine can help us tell us about what happened in the kidneys is it helps us to figure out clearance, okay? So the clearance rate of a particular solute, X, is the volume of plasma that gets quote unquote cleared of that solute X per unit time, okay? So that might not make sense to you right now, but we're gonna go over it over the next few slides. Basically, it's a representation of the rate at which we are excreting a particular substance from the body, okay? So to illustrate the concept of clearance, we're gonna look at this figure right here. Just to orient you, this is our vasculature. We've got our afferent arteriole, our glomerulus, efferent arteriole, and peritubular capillaries here. And then we've got our tubule here. So we've got Bowman's capsule here and the whole rest of the tubule down here, okay? What we're gonna be doing is we're gonna be looking at this box right here that represents 100 milliliters of plasma, okay? In that 100 milliliters of plasma, we have 4 molecules of this substance called inulin. Don't worry, like for now, don't worry about what inulin is, just suffice it to say there's this molecule called inulin, and we have 4 of them in this 100 milliliters of plasma, okay? And what we're gonna do is we're going to follow what happens with, basically we're gonna follow this 100 milliliters and see what happens to it as it moves through the kidney. All right, so we're starting with our 4 inulin molecules in 100 milliliters of plasma. That 100 milliliters is gonna move up here and it's going to filter through to Bowman's capsule, okay? So in this example, our GFR is 100 milliliters per minute, which means that 4 inulin molecules are being filtered into Bowman's capsule per minute, okay? So inulin is going to be present at the same concentration in the tubule that it was here in the bloodstream, because remember, the filtrate is going to have basically the same composition as the plasma here, except that we don't have proteins. And inulin is not a protein, so it's going to be present at the same concentration, okay? So we followed our 100 milliliters, okay? Now it's here inside the tubule, and now it's filtrate instead of plasma, but it has the same concentration of inulin, these 4 molecules. Okay, All right, now let's follow what happens to our inulin or to our 100 milliliters. Okay, our 100 milliliters is going to move on down the tubule. Okay, as it does that, what's going to happen is we are going to reabsorb fluid from the tubule, right? So remember that almost all of the fluid that filters into the tubule gets reabsorbed. Okay, so for the purposes of this illustration, we're just going to say that the entire volume gets reabsorbed. So we have 100 milliliters here. It's going to get reabsorbed as 100 milliliters here. However, inulin is a molecule that doesn't get reabsorbed. So basically what we've done is we've reabsorbed our 100 milliliters of fluid without reabsorbing the inulin that went with it, okay? So reabsorb 100 milliliters, leave 4 molecules of inulin in the filtrate. At the end of this whole process, we are going to end up excreting all of that inulin. So that's 4 inulin per minute, okay? And then basically we end up with this 100 milliliters of fluid here that has been, basically it has had its inulin removed from it. Okay, so it's kind of like saying, we say that we took our 100 milliliters, right? So we started with 100 milliliters with our 4 inulin molecules in it. At the end, we ended up with that same 100 milliliters, but without inulin in it. Okay, so we say that we cleared that 100 milliliters of inulin. Okay. And since we have, since our GFR is 100 milliliters per minute, then we are clearing 100 milliliters per minute of inulin, okay? Another way to think about it is, I don't know if it's another way to think about it, it's kind of the same thing, but so you can say that we excreted 4 inulin per minute from our body. That 4 inulin, based on its concentration, corresponds to 100 milliliters. Okay, so it's like saying we took that 100 milliliters and we cleared it completely of inulin. So we say that the clearance of inulin is 100 milliliters per minute. Okay.